All
Go ahead.
righty. Well, it's my pleasure to introduce Dawn Blitz today. Dawn joined my lab as a first year grad student back in 1992. So I just discovered that this makes year 30 that Dawn and I have been together. And I must say, in a Dawn-free world, my PubMed pages would be substantially downgraded since we've co-published 18 very nice papers over that time. Dawn embodies all the attributes one hopes to find in a colleague. She's passionate about her research and about science in general. She's super motivated, as is clear from the fact that she continues to be productive in the face of quite a hefty teaching schedule. She's smart, skilled, and a first-rate trainer, as is evident from the success of her trainees. She's a wonderful collaborator who always holds up her end of the bargain. And collectively, we owe her a considerable gratitude for her continuing service to our community, because she keeps on planning our annual STG meeting and keeps on running the STG cycle at the Neural Systems course in Woods Hole in the summer. So thank you, Dawn. And without further fuss, the stage is yours.
All righty, let me share my screen. Okay, thank you very much, Mikey. That was a very nice introduction. I'm glad that Nellie told you had to be nice. Thank you to Smita and Nellie for the invitation to talk today. I'm definitely excited to discuss research with you all today. And along those lines, one of the first things I want to do is to just actually say thank you to the STG and Extended Small Circuits community. This is an amazing group, and I'm incredibly grateful to be part of it over all these years. You've all continue to be amazing examples of how to do just incredible science, but also terrific educators and mentors, and just really generous with supporting other people in the community. So I really appreciate that. And I tried to come up with a way to sort of encapsulate what the community stands for, what we represent. And so I tried STG first, and I came up with science through generosity. And generosity maybe wasn't I don't know, it wasn't quite set on that. So then I tried STNS, and I thought science, teaching, nourishing the fashion sense was apropos. And so, as I said, I just appreciate being part of this community and the support and collaborations that I've had and hope to continue to have. And along those lines, I also want to say thank you to the STG cycle folks. So this is, as Mikey said, part of the Neural Systems and Behavior course at the MBL. A 1994-ish, not sure what year that is, Mikey started the STG cycle. And I'm also incredibly grateful to have been part of it for so many years. So going back to Woods Hole for 2 weeks and over many, many summers, not only is really great for me because it kind of replenishes my soul to get back to the ocean, but it also allows me to interact with so many of you, work side by side with you. And I've learned so much about STG and neurophysiology and teaching and mentoring, and maybe even had a little bit of fun along the way. So again, thank you to this group as well. I also will give a shout out to my science big sister, Melissa Coleman. She did a good job of teaming up with Mikey to try to toughen me up, especially in the beginning, and continues to be a source of support and friendship. And of course, I need to thank Mikey, who let me in the lab not once, but twice. And he took my assignment that he was my mentor for life very seriously, and has really done an amazing job over the years. So this includes many, many excellent meals. He's done a fantastic job at teaching me how to deal with people in the lab. And his advice over the years has always been really very straightforward. He doesn't mince words. And I'm I'm really grateful for his continuing mentorship and friendship throughout the years. And he's not done being my mentor for life quite yet. So I will just say, thank you, Mikey. It is good. All right, so why did I get to STG? Well, I went to grad school relatively naive. I wanted to figure out the nervous system, but didn't really know what that meant. And so my first year in grad school, Mikey was teaching a class and he was talking about central pattern generators. So this included work from Ann Beckhoff's lab and talking about how folding the chick's head under its wing and tucking it back into a fake egg could reinitiate hatching, most likely from the same circuitry that allows them to walk. And I thought central pattern generators, these flexible circuits sounded amazingly cool, and I wanted to learn more about them. And then I was scheduled to start my rotation in Mikey's lab in January. So he let me borrow a copy of the Green Book, the first STG book, over the holidays. And I still remember looking at these two figures here, which are called the electronic equivalent diagram for the pyloric and gastric circuits. And just really falling in love with thinking about the nervous system as electrical circuits. And in this case, having the cells have their own names and then trying to tease apart what all these connections do to let us produce different versions of behavior. I was really hooked before I even started the rotation. In addition to this, I would say some of the things that have kept me or brought me back to STG are the fact that I really love trying to sort out how the changes in the cells and the synapses allow us to select different outputs from a circuit. But I really like doing this and asking these questions in the context of a rhythmic circuit. So of course, this crowd knows that the isolated stomatogastric nervous system gives us this amazing cellular access, the projection neurons, the circuits, the muscles, sensory inputs, all of this can be in the dish. And as I said, that's really one of the things that I most enjoy and appreciate about working in the stomatogastric nervous system. So work I'll tell you about today, with one minor exception, was all in Cancer borealis, that crab shown there. And the first time I joined Mikey's lab, they were in the process of asking whether bath application mimics neuronal release. And some of this is a little bit of a summary from Mikey's talk, but it was a year ago. So just bear with me. Hopefully it's not too redundant. So of course, Mikey, when he was a postdoc with Eve, showed that the Michael Philip Nussbaum neuron or modulatory proctolin neuron releases proctolin, excites the pyloric circuit, illustrated with these schematic sticks down here. And the effects on the pyloric circuit are really similar to those of bath applying proctolin. So the initial answer was, yeah, bath application can be quite similar to neuronally released modulator. But then in his lab, Melissa Coleman, Brian Norris at the time, were working to identify additional projection neurons. And again, this is in the crab. In Mikey's talk, he discussed a lot of the previous projection neurons that were identified in other species. So I'm going to leave that to his talk. So what they found is that in addition to the MPN proctolin containing neuron, MCN1 and MCN7, the Melissa Coleman neurons, also contain proctolin. And it turns out that even though they contain the same neuromodulator, they elicit different activity from the networks. So the pyloric effects are different. There's activation of the gastric network as well. And in part, this is because there are co-transmitters present. And I'm not going to go into some of the details of other reasons that these neurons have different effects. But co-transmitters were certainly a component of that. So for my dissertation work, I focused some more on MPN And MPN not only projects to the stomatogastric ganglion, but it also sends its arms out to the commissural ganglia, which allows it to interact with some other projection neurons. And you're going to hear about these quite a bit today, MCN1 and commissural projection neuron 2, or CPN2. So these are two projection neurons that activate the gastric mill rhythm. And what we found is that MPN inhibits these two projection neurons up here in the commissural ganglia. That removes their activation or their excitation of the gastric mill rhythm. And so MPN indirectly inhibits the gastric mill rhythm. And we knew that Proctolin mimics MPN's effects on the pyloric circuit. And so I asked whether Proctolin also mimicked its inhibition of these parallel pathways. But bath-applied Proctolin showed that it actually excites CPN2 and MCN1. So in this case, bath-applying the modulator had the opposite actions from the neuron. And I found that instead, MPN uses GABA to inhibit MCN1 and CPN2. And so here we have this modulatory neuron that directly acts on one circuit via its neuropeptide. So bath-applying that peptide mimics those actions really well. But it inhibits these competing projection neurons with a co-transmitter. So bath-applying the peptide does not mimic those effects. And this allows the neuron to indirectly inhibit the second circuit. And so I'm going to keep using this general schematic here as I go through the talk to kind of summarize things. So one of the big takeaways from this work in Mikey's lab was that bath-applied neuromodulator does not necessarily mimic neuronally released modulator. And one of the key reasons for that is the presence of co-transmitters, which of course are incredibly common. And we know that there's tons and tons of them present in the STNS and all other systems. And then one other thing to come from this work is that the function of co-transmitters can be spatially segregated. And Mikey's lab has gone on to do some really nice work also at even at individual arbors at how different co-transmitters can be separately regulated. All right, so this is kind of a really super quick summary of work in Mikey's lab. And at this point, when I was graduating, I thought that it would be a good idea to kind of push myself and experience some other things. And so I started out with a postdoc in a vertebrate central pattern generator. I went to work with Nino Ramirez when he was still at the University of Chicago. And we heard a beautiful talk from Nino a few months ago. So there I was looking at the respiratory CPG in a mouse brainstem slice. And using this preparation, I was looking for an in vitro model of an in vivo phenomenon where the respiratory network has a really long-term adaptation to brief repeated episodes of lowered oxygen or anoxia. And this really gave me a good experience in a vertebrate CPG. And Nino was very patient with me coming from STG and missing my giant cells. And he gave me a few nudges about how they had beautiful pacemakers in the respiratory CPG that were really very similar to things like the PD neuron. So after this experience, I decided to push myself even further out of my comfort zone. And I went to work with Wade Regehr at Harvard Med School to look at synaptic plasticity. And so in this case, I was again using a mouse brain slice preparation, but now looking at sensory pathway, so the retinal ganglion cell synapses onto thalamic neurons, and looking at how the strength of synaptic currents is really finely sensitive to the timing of the inputs, and then how that plasticity regulates the way that the thalamic neurons are responding to this incoming sensory information. And so this was sort of a Goldilocks and the Three Bears, where I went to the respiratory CPG and I looked at long-term plasticity. And then in the thalamus, I was looking at short-term plasticity of synapses, and then decided to go back towards the middle, looking at neuromodulation on a sort of intermediate time scale, but also returning to the stomatogastric system, again, because I wanted to ask about modulation of circuits, but in this context of a more intact system. And so when I came back to Mikey's lab, I moved kind of one step up and started to look at how extrinsic inputs into the system were regulating the activity of projection neurons. So this was, again, kind of good timing for me as I came back to the lab, because Mark Beenhakker, through his dissertation work, had very nicely described this population of mechanosensory ventral cardiac neurons, or VCNs. So they're neurons that are located at the midline in the cardiac sac. They are activated by stretch of the cardiac sac region, activate projection neurons, which then elicit a particular gastric mill rhythm that I'll call the VCN gastric mill rhythm. And then just a little before I returned, Farzan was in Mikey's lab doing a sabbatical, and along with Sherry Sederman and Rachel White, they were finding that there's this very short little commissure, and the schematic really doesn't do this justice, but they found that the short little commissure, they could stimulate it. And as we collaboratively described, it allowed us to stimulate this axon bundle that terminates in a club-like structure. And I'm going to come back and talk about that club structure later. The point for now is that these other neurons, called the POC neurons, they activate the same two projection neurons, which act on the STG circuits, but elicit a distinct gastric mill rhythm that I'll call the POC gastric mill rhythm. And so let me just briefly review the difference between these two gastric mill rhythms. So here's recordings of the two projection neurons you're going to hear a ton about, CPN2 and MCN1, LG to monitor the gastric mill rhythm, PD for the pyloric rhythm. And the unique thing about the POC gastric mill rhythm is that everybody, the two projection neurons and the LG neuron, have this pyloric timing, right? So their activity is interrupted in time with the pyloric rhythm. And this distinguishes it from the other version, the VCN gastric mill rhythm, because in this case, although during retraction, so in between the LG bursts, MCN1 and CPN2 do have pyloric timing, but during the LG burst, which is the protraction phase, the LG neuron fires tonically, and so do the two projection neurons. Okay, so the key distinction is that they lose their pyloric timing during one phase of this VCN gastric mill rhythm. And I'm going to use these schematics going forward just as a reminder of which rhythm I'm talking about. And I should also point out that these distinctions between these two rhythms, which initiate in the projection neurons, is carried all the way through. So the motor pattern is different, the muscular activity is different, and the behavior is different. And so some of this work is from Mikey's grad student, Rachel White, and then a collaboration between Mikey and Wolfgang's lab with Wolfgang's student, Florian, actually putting an endoscope into the crab's stomach and stimulating these pathways. And so in the POC rhythm, the LG neuron is controlling the lateral teeth, and they're coming together in kind of a chattering fashion. But in the VCN rhythm, they showed that the lateral teeth come together smoothly and they generate greater force at the midline. So we've got two different activities in the same two projection neurons that is carried through all the way to a behavioral difference. And so I wanted to address how it is that we get these two different types of activity in the projection neurons. And the obvious difference is the difference in the patterns of projection neuron activity. I'll come back in a little bit to also talk about differences in the firing rates. So I'm going to start with the activity patterns. And this means that I get to add in my favorite arrow in this schematic here, which indicates that there is feedback from CPGs to their projection neuron inputs. And this is really any CPG that I know of, any CPG that's been looked at, but it's also really common, of course, in other networks throughout the nervous system. This is sometimes referred to as an efference copy or a corollary discharge. I know, Abdel, I don't know if you're here, you've asked in the past about efference copy in the system. What it means is that sort of a report of what the motor activity is at that time is sent to other regions of the nervous system. And the consequence for projection neurons is that the projection neurons, and these are two examples from the European lobster, CP and P, their activity is periodically interrupted in this case in time with the ongoing pyloric motor activity. So here they showed that in this example, the P cell has its pyloric activity, and that if you block this interneuron, well, they killed it with photoinactivation, then you see that the P cell switches from having that pyloric timing to being tonic. And the same is true in Borealis. So here is an example of MCN1 where it has this pyloric timing. And what's been shown in several papers is that the AB neuron, which does, of course, project up to the commissural ganglia, synaptically inhibits both MCN1 and CPN2, so that when you remove feedback from the networks, you get this tonic firing in the projection neurons. So if we think about what I've shown you with the two different projection neuron patterns, the question then is, why does the pyloric timing go away physically, right? How come during each protraction phase, when LG is active, the projection neurons lose that pyloric timing? And I can tell you that it's not because AB is turning off. So this is just a recording of AB in the SON. And during both retraction, when there is pyloric timing in the projection neuron, and protraction, when that pyloric timing goes away, of course, the pyloric rhythm is ongoing and you can see AB is firing its activity. So what is happening? And for this, I brought in my postdoc experience. So, we were used to looking at it in current clamp and looking at the impact of the feedback synapses. But in Nino's lab, a number of people were looking at these beautiful barrages of inhibitory currents and how they were rhythmic barrages and how they were modulated. And then, of course, in Wade's lab, I learned voltage clamp and analysis of synaptic currents. And so when I was coming back to Mikey's lab, this is one of the things that we talked a lot about, is that bringing a voltage clamp and analysis of synaptic feedback. And so what you can see, it's a little bit compressed here, but during each AB-PD burst, recording from MCN1 and voltage clamp, there are these really nice barrages of summating inhibitory postsynaptic currents. Okay, and this is what I'm going to look at in the next slide. So here's MCN1 and voltage clamp, rhythmic AB inhibitory currents. And what you see is that in this VCN version, when LG fires, which is the time that the pyloric feedback is eliminated from the projection neurons, the synaptic currents are eliminated. Okay, so there's this phasic synaptic currents are on and off during protraction versus retraction. This is only for the VCN version though. So this regulation of feedback is actually state dependent. In the POC version, the pyloric timing persists and the synaptic currents are present during both phases of this particular version of the rhythm. And so this suggested that there's some kind of local regulation of AB up there in the COGs where it's synapsing onto the projection neurons. And to address this, I took advantage of wizard Ron Calabrese's description of how to control multiple spike initiation zones. So from this 1980 paper here. And so as you know, there are many neurons that have more than one spike initiation zone. And in rhythmic neurons, there are multiple rhythmic zones that are typically controlled by one primary one. And so here I'm recording from AB near the entrance to the STG, as well as up in the SON. And under normal conditions, the primary zone is near the STG. AB oscillates and it generates action potentials. They propagate towards the COGs, and so they occur later in this SON recording. But hyperpolarizing the primary site to turn off this spike initiation zone enables another zone that's also rhythmically active up in around the COGs to be activated. And so now you see the spike initiates or happens in the SON recording first, and then comes down to my STG recording. So it would have been nice, of course, to get into the AB terminals and really look at what was happening there. But this is kind of a way of sort of viewing what's happening to AB up in the COGs. And so here in the control condition, the action potentials are initiating the STG. So they're riding off of these oscillations. And AB action potentials occur during both protraction and retraction. But when I hyperpolarized AB in the STG, you can see now there's no oscillations, but there are these rhythmic bursts of action potentials. And the rhythmic bursts are inhibited during each LG burst. So this means that locally within the commissural ganglia, something is inhibiting AB presynaptically. And I have some other evidence to support the presynaptic part of that, that results in the feedback from AB onto the projection neurons being eliminated during that protraction phase. Okay, so this is one of my favorite projects. I really love this project because partly because it was challenging and I find it really interesting. It was a long-standing mystery in Mikey's lab. Why does the pyloric timing go away? But I also really like it because it finally gave a home to this N of 1 that has been on Mikey's bulletin board since 1996. And this was an accidental kind of crappy recording of AB's axon that I made. And because it was relatively crappy, I accidentally inactivated the primary spike initiation zone. And so you can see that the action potentials in AB are kind of coming at random times. So they are initiating in the COGs and coming down. And what we saw is that when MCN1, so the middle-sized unit here, when it was receiving pyloric timing, there were action potentials in AB. And when it was not receiving timing, those action potentials disappeared. So it took quite a few years to finish that story. This poor little N of 1 had a long wait, but it was nice to finally bring that to a conclusion. And the conclusion, as I've shown you, is that when the VCN pathway has been activated, something happens, and there is now phasic inhibition of AB up at its terminals. And that only occurs during one phase. So that eliminates the feedback inhibition during the protraction phase. Presynaptic inhibition is relieved during the retraction phase so that the feedback is effective. And this presynaptic inhibition is state dependent. So it does not occur during the other version, the POC version of the rhythm. And that allows the pyloric feedback to be effective continuously. Okay. So I think that the feedback from the motor circuit to its projection neurons was such a standard and was so well established that it hadn't really been considered that it might be flexible. So this was an initial indication that there can at least be some regulation of the CPG feedback to its projection neurons. But this was fast phasic regulation, right? I'm really interested in neuromodulation. And so I wanted to get at whether there might be other ways that the feedback might be modulated. And if we consider these projection neuron recordings, when we initially impale them, what we see is that there really isn't very obvious pyloric timing. And one possibility is because they're just, they're resting at around -60 or so. But even if I just depolarize the projection neurons, yeah, there's some pyloric inhibition, but it's not really that pronounced. However, after stimulating one of the pathways, like the POC pathway, you can see that the strength of the inhibition looks much stronger, and it has a more, it's more effective at inhibiting the activity. And so this suggested that the AB feedback might actually be modulated. So again, if we look at the synaptic currents, and now you can see this is just one cycle of AB activity. And you can see these really pretty iPSCs from individual AB action potentials that summate. But the point here is that prior to POC stimulation, in either MCN1 or CPN2, the feedback is relatively weak, but the synaptic currents are enhanced after the POC stimulation. And this is a long-lasting effect. This is a plot of the area, so just integrating the area of this cumulative current here, the summating currents, and going from the time after the POC stimulation, you can see that the amplitude increases and stays increased, and that's for at least 15 minutes, which is important because that's the duration of the POC triggered gastric mill rhythms. One thing I'll just say, I won't show the data for this, there was no change in AB activity. And in fact, you can count that there are 6 iPSCs here and 6 iPSCs here. There's no change in firing frequency either. So this does seem to be an enhancement of the synapse and not just of the, not just a change in AB's activity. So I then wanted to address why the feedback was being enhanced. How necessary is this? And so with some help from Farzan, I used this dynamic clamp setup here, where I recorded from MCN1, hyperpolarized AB, which of course means I hyperpolarized the 2 PDs, and then the electrical coupling shut off AB. In the software, we created a model AB neuron, so I could then control the strength of this artificial feedback synapse. So no feedback synapse, MCN1, the projection neuron fired tonically. But in the presence of the feedback, using that model cell, I can replicate that nice pyloric timing. So to address whether the modulation was necessary, here you can see the effect of the natural AB feedback on MCN1. And then if I use the control amplitude, so this is the weaker amplitude from before activating that POC pathway, again, there's some regulation of MCN1's firing, but it's not really having much of an effect. And then if I instead use the modulated amplitude that occurs after stimulating that POC pathway, now you can see there is this really nice interruptions in MCN1's activity. And so the influence of the feedback synapse is kind of doing its job now. So this indicated that the feedback actually needed to be enhanced. So that means the baseline strength of feedback in this motor system isn't strong enough to do what it needs to do. There needs to be this modulation to enable the projection neuron activity pattern to be correct. And this extends out to the motor activity. So here I'm now using this activity pattern in MCN1 in response to the weak control feedback as a stimulus, and looking at the motor activity in LG. The up arrow or the arrowheads indicate when there should be gaps in LG's activity, and they're not really there. But the MCN1 activity that is driven by the enhanced feedback does nicely interrupt the LG pattern. And so we need to actually enhance the feedback in order to get the appropriate activity pattern at the level of the projection neuron and at the level of the motor output. And remember, this is necessary for those teeth to have the chattering movement instead of that strong, smooth movement. Okay, so what that means is that this extrinsic pathway not only has some direct effects on the projection neurons to increase their activity, but it also has this long-lasting modulation of the feedback strength. And in this case, this is important for regulating the activity of the projection neurons and regulating the pattern of activity. And I think everybody's really good about this, but I'll just say, please interrupt if you have questions along the way. Okay, so now I've shown.
Ron, I have a question.
Sure.
I remember when you did these long-lasting experiments with the respiratory network, it drove you crazy that it was individually so variable. Now, when I saw your long-term change, it was also pretty variable. I mean, like, did you see similar things, and did it drive you crazy too, or not?
Variability always drives me crazy, and it's currently driving me crazy in the gastric mill rhythm as well.
Okay.
Yeah, I mean, you're right. It's definitely variable. I think some of it has to do with, you know, the crabs have been sitting in the tank for a couple weeks before we take them. Sometimes they eat the food they're given and sometimes they don't. And I don't know what was going on with them before we got them. And so we'll often see that the baseline is variable to begin with.
Yeah.
And so then I think, depending on what it's already experienced, the effect that any particular input is going to have on it is then variable on top of that. But yeah, it definitely,
cool.
It is frustrating.
No, I'm glad that the STG is not a machine, of course.
Thanks for the question. Okay, so now I've shown you that there is phasic regulation and there's long-term modulation of feedback. And so far, these two aspects are important for the pattern of the projection neuron activity. But of course, there's also the activity rate that can be really important, especially when we're thinking about neurons that release neuropeptides in addition to small molecule transmitters. And so I next turn to looking at how what it is that might regulate the difference in the rate of activity between the projection neurons between these two scenarios. And this is my reminder that this was the point that I flew the coop and moved from Pennsylvania out to Oxford, Ohio to start my lab at Miami University. So for this, I'm going to really skim this and kind of hit the highlights of it. But for firing rate, I'm going to focus on the CPN2 projection neuron because it was only its rate that was different between the two versions of the gastric mill rhythm. And I'm going to focus on the VCN rhythm. So this is the one, of course, where the projection neurons fire tonically during protraction when LG is active. And in this plot of instantaneous firing frequency, you see that CPN2 goes up to about 40 hertz. But then when I eliminated feedback, it fires relatively tonically, and the average rate is about 20 hertz. So it was really a big, and that was consistent. It was about half the frequency that it was during protraction in the absence of feedback. So I have to give you a little reminder that there's the second feedback neuron, interneuron 1. So this is part of the gastric mill rhythm generator. And it also, of course, projects into the stomach commissural ganglia. And Brian Norris, when he was with Mikey, showed that this forms an inhibitory synapse onto CPN2. It's, again, a fast inhibitory synapse. In the previous slide, I showed you that if I block all the feedback, then the CPN2 firing rate drops. And I'm going to skip the data, but tell you that if I looked at AV versus INT1, it was only INT1 that was regulating CPN2's firing rate. So that's the one I'll focus on here. And again, this is the VCN version only. So when Brian looked at the feedback onto CPN2, he actually did it in a modulated condition. So they were looking at in the presence of a muscarinic agonist. But when I looked at INT1's effects on CPN2 in control conditions, it really didn't do anything. So here's when INT1 was active, no obvious effect. And then after stimulation of this VCN pathway, now when INT1 was on, you saw this really nice inhibition. And again, that's evident in the synaptic currents. So in control, there's really no obvious INT1 synaptic currents in CPN2, but after stimulating the VCNs, there's this nice big inhibitory current. So we have now another feedback pathway that's modulated. What's the purpose of this one? And so I'm going to just kind of briefly summarize that. In the control condition, the INT1 neuron doesn't really do anything to CPN2, but particularly in that VCN version of the rhythm, this synapse is greatly enhanced. So you can see here, INT1 is always active in retraction, and it has a strong inhibition of CPN2, and that triggers a rebound burst. So CPN2 now fires at a higher frequency than it does just to the VCN excitation. So this strong frequency at 40 Hertz, this high firing frequency of 40 Hertz, is post-inhibitory rebound from INT1 inhibition on top of the VCN excitation. Okay. And what this means then is that now this VCN pathway, or another pathway, has direct effects on the projection neurons again, but the way that it is sculpting the activity of the projection neurons is by modulating a different feedback pathway, INT1. And through these interactions with the intrinsic properties, it is regulating its activity rate. And one thing I forgot to say here, I looked at PIR, that property itself in CPN2, and the rebound property in CPN2 was not different. The only thing that was different was the strength of this inhibitory feedback synapse. So as I said, the inputs to the projection neurons, it seems like all they're doing is getting the cells activated. And then what's sculpting their activity levels and their activity patterns is the way that feedback pathways are regulated, which I thought was a really interesting and kind of unexpected result. And for this project, I have to say a very special thank you to Nellie Dower, because she flew out to Ohio for spring break. And our big exciting spring break was that she stayed in a very unfinished bedroom in the house. And we spent really long days in the lab. She did lots of dissections and helped me with axon recordings. And this was really pivotal to me completing this project. And the timing was important because it meant the paper was published before I submitted my tenure package. So thank you again for that, Nellie. All right, so I've shown you how feedback, or the regulation of feedback, can kind of sculpt the projection or an activity, which is then critical for the output we get for the actual behavior. And one other consequence that I'm not going to have time to talk about is now these projection neurons actually respond differently to other inputs. So GPR is a sensory neuron. So this was first described by Paul Katz with Ron Harris' work. And then in Mikey's lab, we showed that GPR excites these two projection neurons. But Mark Beenhakker, as part of his thesis work with Mikey, showed that in the VCN modulatory condition, this state, that excitation of the projection neurons from this sensory input is blocked. However, in a collaboration between my lab and Mikey's lab, we showed that in this modulatory state, the GPR sensory excitation of the two projection neurons is intact. So this current state of feedback pathways to projection neurons appears to be able to also regulate how they respond to additional inputs. Okay, so I'm going to take a jump to the side here and actually talk a little bit about an anatomy project instead of physiology. And as I said earlier, Mark in Mikey's lab told us that these VCN neurons are mechanosensory. So they appear to be activated when the cardiac sac stretches with food, and then they activate their version of chewing rhythm. The POC neurons, as I'll talk about, we know a ton about their terminals, and we have no idea where they come from, where they originate. So this is something that I actually started working on as the very first thing I did in Mikey's lab, and I'm still working on it. So some of you may remember that before they were called the POC axons or POC neurons, they were called the club, because this first paper in '88 used substance P immunoreactivity to identify this club-like structure. And then my very first project with Mikey, we looked at a locust-related peptide. So these are all in the tachykinin family. And again, the immunoreactivity looked like this club structure. Andy Christie is a postdoc with Mikey in collaboration with Dick Nossel. Finally identified the native peptide, which is Cancer borealis tachykinin-related peptide 1A or CABTERP. And then, of course, the work, oh, right, so one other thing here, Andy Christie led this group here to determine that this structure, this crazy looking blob of axon terminals, is actually an endocrine structure. So it wraps around hemolymph lacunae, enabling the CABTERP to be released into the circulation. And at that point, they renamed it the anterior commissural organ, or ACO. And then the paper we had from this collaboration among all these people in Mikey's lab showed that there's also paracrine actions, because the CABTERP that's released from this endocrine organ has these local effects on the projection neurons. And I also have to show these pictures because I still absolutely love these pictures. This is confocal work from a grad student, Yvonne Swally, and undergrad Lexi Monti. And what's shown here is a confocal 3D rendering of confocal Z-stack, where this is a fill of MCN1, the projection neuron, and then in red is the immunolabeled club structure. And I'm still amazed. I still think this is beautiful, how MCN1 just really wraps around the endocrine organ. And this is one example, and this is just another example where they really wrap around each other. But what this means is that we know tons and tons about, maybe not tons, but we know a lot about the axon terminals, but we don't know where these cell bodies are and where these axons originate. And so if anyone needs a little reminder about the rest of the crustacean central nervous system, we have the supraesophageal ganglion, or the brain, kind of behind the eyes. The COCs come down, and the COGs, the commissural ganglia, are little out pockets of those. And then the stomatogastric nervous system kind of projects posteriorly, of course, wrapping around the foregut, which would be about here. And then there's a really long distance that the COCs travel and enter into the thoracic ganglion way down in the ventral, way down in the belly of the crab. And thanks to Amanda Rainey for adapting this, this image for us. And in the 2008 paper, we showed that the axons of the ACO, or these POC axons, go backwards to the thoracic ganglion, and that's where we stopped. And then I've had several undergraduate students, as well as Amanda Rainey, a grad student who spearheaded this project, showed that once the POC axons get into the thoracic ganglion, they form what we call the central tracts. So they actually go all the way around where the artery projects through. So they make it all the way through the thoracic ganglion, and they have branches into every neuromere, all the leg neuromeres, the antennal neuromeres, the abdominal. And Amanda painstakingly went through the whole ganglion and found about 100 cell bodies, none of which connect to these axons. So it looks like the POC cell bodies are not in the thoracic ganglion. But when she looked more closely with her amazing confocal skills up here, when the POC axons come in, they turn. And then this is a 90-degree rotation. So you can see the axons coming in, and they branch and they make this turn here. And what happens is, like, so here they're coming in from the commissural ganglia. They come into the TG, the thoracic ganglion. They immediately turn and head right back out another nerve. And I was the one doing most of these dissections and got kind of grumbly about how big borealis is. So I did a few of these in Carcinus manus, just because it's a smaller crab. And overall, it seems like these nerves, there's of course bilateral, go all the way back to the mouth, or the mouth area, the mouth parts. And I think it's the mandibular nerve. And so I didn't know the names of all the mouth parts. If you don't, the mandibles are those smooth, hard structures that you have to kind of pry apart to see the opening into the esophagus. And so what this means is that the axons of these POC neurons, if we go backwards, all the way down to the thoracic ganglion, they project throughout the whole thoracic ganglion, go out every leg nerve, and they do bifurcate. So we have that data, at least some of them bifurcate, and they go all the way up this mandibular nerve to the mandibles, which, remember, are really close to the CRGs. So in Borealis, at least, this is like 3 centimeters down. Who knows how far they go out these leg nerves, and then 3 centimeters all the way back to almost the exact same area in the animal, which I think is just crazy. But the sad thing is, I don't know where the cells are still. So I don't know if they're in the mandibles. I haven't had luck up there yet, although I've had maybe some hints. They might be out somewhere in the legs. And so Mikey calls this my white whale, and I think he's right, because I started on this in 1992. But if we put it into a functional context, these are the neurons that activate MCM1 and CPN2 in this manner, and LG, and elicit that chewing motor pattern. And so I really want to know where they are and what it is that initiates this particular version of chewing. But we don't know yet. All right. So if there's no questions at that point.
Yeah, I want to ask you a question. So do you have any idea if there are ganglia up there, up near the mandibles, or?
No, there is a mandibular receptor organ that's sort of, in this area around some of the mouth parts, but it doesn't seem like that's where they're coming from. So my guess is that they're sensory, sensory neurons that are maybe on some of the little hairs on the legs, or I mean, I want them to be in the mandibles because it's a smaller target. But yeah, I don't know. I think the mandibular receptor organ is about the only option up in this area. Once you head out the leg nerves, I have no idea. Okay, so some of you may be saying, All right, that's enough with MCN1 and CPN2. How about some other cells? So I'm just going to really briefly talk about a few other cells. And I'm going to keep it really brief because this is recent work that my grad students have been presenting at STG meetings. So Savannah and Ryan, I'm sorry, but I'm going to really skim over this stuff. Okay, so this all started one year, I don't remember which year, but Patsy Dickinson and I sat on the carpet upstairs in some convention center, and she spread out a bunch of pieces of paper to show me recordings, to show me what this new peptide, gly-1-cifamide, was doing. And the thing that stood out to me was an excitation of LPG. And many of us don't really think about LPG too often. But I knew we had some information about this. So we turned to Mikey and his many, many boxes of chart recorder paper. And this is just one example. And I'm just using it to say that this particular projection neuron MCN5 really strongly excites LPG. And it actually elicits gastric timed bursting, which is kind of unusual, right? Because in Cancer borealis, LPG is part of this pacemaker ensemble. So let me use different recordings to make this point a little bit more. First, Patsy and Mikey and I formed a collaboration along with Andy Christie. And the first thing we did was just to determine whether MCN5 contains gly-1-cifamide, which it does. So this is MCN1, which does not have the gly-1-cifamide, but MCN5 does. So after all these years and after all this time of talking to you today, I'm kind of back to the original question of whether BATH application mimics neuronal release. So the original description of gly-1-cifamide in Cancer borealis was this collaboration among our 3 labs. And then Savannah picked up this project as it moved into, as we moved on with it some more in my lab. So the top is a set of traces in saline, so recording the LP neuron. The gastric mill's off, so there's no DG activity. But LPG is firing in time with the PD neuron, right? It's purely a pyloric neuron here. Then she stimulated MCN5, and it did a few things, including activating a gastric rhythm. So here you can see some DG bursting. And it switched the LPG neuron from purely pyloric to both pyloric and gastric. Right? And so what this means is periodically LPG escapes from the pyloric rhythm to fire these longer duration bursts. And we found that gly-1-cifamide mimics some, but not all of the MCN5 actions. Okay, so here on the bottom is gly-1-cifamide, the same preparation as the top. It activates bursting in DG, so that was good. It does excite LPG. We kind of get these envelopes of enhanced activity, but you can see that the bursts are extending beyond the PD burst, but it's not fully escaping from the pyloric rhythm. So that was a difference. And then the other difference was that LP was inhibited by MCN5, but excited by the peptide cifamide. So I'm going to skip this data, but just tell you that Savannah showed that MCN5 actually inhibits LP by using glutamate. So it's a fast glutamatergic inhibition. So again, co-transmitters are important reasons why BATH application might not mimic neuronal release. But we know that LP inhibits the pacemakers, including LPG. And so we reason that maybe this difference, maybe it's because it's not inhibiting LP, this prevents the peptide from enabling LPG to fully escape the pyloric pacemakers. And so what Savannah did was to apply gly-1-cifamide. LPG bursts are extending beyond PD, but then as soon as she hyperpolarized LP, you can see that the LPG neuron now fires these nice longer duration gastric time burst that escape from the pyloric rhythm. So as long as we inhibit LP, and she moved from hyperpolarization to photoinactivation, but if we add that in, then gly-1-cifamide is a pretty good mimic of MCN5. And so we've gone forward using that as a model for MCN5 activation. Okay, so we were really interested in this switch of LPG from single frequency to dual frequency. It's not the first time this has been seen. So Jim Wyman's paper with Eve in '91, where he stimulated the COC, showed LPG making a similar switch. And we don't know, but Mikey and I like to pretend that this is actually activating some input that then activates MCN5, but we don't know. Okay, and so this kind of, I want to just take a really brief minute here to talk a little bit about how mysterious LPG is, because in the lobster, LPG is pretty much a gastric neuron. And so this is just an example of its activity in time with the DG neuron. But in Cancer borealis, it's primarily pyloric. And it looks so much like PD that initially in Scott Hooper's paper with Eve, they called it PD-in for PD intrinsic because it innervated some intrinsic pyloric muscles. But later, Jim Wyman, again with Eve in '91, showed that it also innervates gastric muscles, and they reassigned it as actually LPG. So I was certainly negligent about this over the years. I think of the dilators as the cholinergic neurons in the system, but LPG innervates both pyloric, both constrictors and dilator muscles. And it turns out that it has retained its cholinergic phenotype. All right, so in the crab, LPG, even though a lot of you leave it out of your circuit diagrams, LPG is part of this pacemaker ensemble. And this collaboration between Dave Schultz and Eve's lab showed that at least for the PD to LPG synapse, electrical synapse, it's rectifying. And I think that's actually going to turn out to be really important in enabling LPG to escape from the rest of the pacemaker neurons. So what we were really wanted to figure out was the mechanism that enabled LPG to do this escape, right? To switch from single activity to dual activity. And neuronal switching, or the plasticity of neurons' participation between networks, certainly not new. In the stomatogastric system, we had lots of examples of neurons changing the network that they participate in or creating new networks. And in many cases, the way that a neuron is recruited from one network and kind of, I think of it as being pulled into a second network, is through a synapse, right? A synapse is enhanced and that kind of pulls the neuron in. But I had an initial hint that maybe LPG worked differently. And so we wanted to look at whether it was actually recruiting itself in a way, whether it was intrinsically generating the burst at the second frequency. There are some examples like Hooper and from Hooper and Milan that you can modulate intrinsic properties to have a neuron be released from a network. And there are also some hints in the respiratory system that modulation of intrinsic properties allows neurons to drop out. But we thought this was going to be a way that intrinsic properties could actually bring a neuron into a new network. And so again, apologies to my grad students for this super quick overview here. This is LPG generating the dual frequency bursting in response to cifamide or MCN5 in the intact networks. And then what Savannah did was to use picrotoxin to isolate LPG from all the glutamatergic input, PD hyperpolarization to eliminate the rhythmic electrical coupling from the pyloric pacemakers. And you can see that this isolated LPG generates these beautiful slow oscillations underlying the slow frequency in the intact condition. And what we called our nail in the coffin experiment Even when Savannah had LPG isolated from both networks and she photoinactivated one of the LPG neurons, LPG still generated this nice slow bursting intrinsically, and it was in fact voltage dependent. So this is an example where the LPG neuron is recruited into the second neuron through modulation of intrinsic properties. And so we think that this may give it a more active role in what it's doing in that second network. And those are things that we're continuing to work on. Okay, so the last two data slides, poor Ryan's data is getting super summarized, but he just did a very nice job of defending his master's thesis on this work. What he found is that there are multiple intrinsic properties that are modulated in LPG. So this is just an example where the post-inhibitory rebound after hyperpolarizing current is enhanced in GLI-1 cifamide. In addition to PIR, he found that the overall excitability measured as a frequency current relationship increased and spike frequency adaptation decreased. And so we think that all of these things are contributing to really like jazzing up LPG so that it can escape from that strong electrical coupling to the other pacemaker neurons. And I hope that this isn't too compressed here. Ryan also used pharmacology to look at potential ion channels or currents that contribute to the intrinsic LPG bursting. And so he's found a role for what looks like IH and a persistent sodium current and a calcium related or calcium current. But in the isolated LPG, none of these currents are individually necessary. So we can still get some LPG bursting when any of these are blocked. But in the dual network bursting, it's almost impossible to get these slow bursts with a blocker of persistent sodium currents. And we can't get these slow bursts if we block when we lower calcium concentration and block calcium or calcium related currents. So I think what this is telling us is that there are these interactions between this fast electrical coupling and the ability of those intrinsic properties to overcome the electrical coupling. And again, that's something that we're continuing to work on. So what I have talked for a long time about is if we are thinking about selecting a particular motor output, bath application of modulators that target that network may not mimic neuronal release. A lot of that has to do with co-transmitters. I've shown you an example where co-transmitter functions can be spatially segregated. Projection neurons are regulated by sensory input, of course, but also the CPG feedback, which is flexible. And that can include a phasic regulation in a state-dependent manner. Long-lasting enhancement of feedback can regulate the pattern and the rate of projection neuron activity through feedback interacting with intrinsic properties. And I think this all goes to show that inputs to the projection neurons may have kind of generic effects, but the individualized actions may have more to do with regulating the feedback connections. And then third, in terms of neuronal switching, these switches between single and dual frequency are really prevalent. And I think recruiting a neuron through modulation of intrinsic properties is a good thing for us to keep an eye out for. And we're really interested in how electrical coupling and intrinsic properties interact. Certainly modeling work from Gabrielle Gutierrez and Eve showed some really interesting complexity there. And that includes a potential role for rectification. And we think that's going to be really important for LPG to escape from the rest of the pyloric circuit. And we're also going to look at its roles in inner circuit coordination. And so thank you to my current students. I have some new students, so I wasn't talking about their data. Fantastic collaborations, funding all my former students, the crabs, and I really appreciate your attention through that long review of things I've been doing over many years. So I'm happy to take any questions if you're not worn out. And this cartoon was just so amazingly close to what often happened when I went into Mikey's office. So I know he included it, and I have to include it as well. So thank you very much.
Nicely done, Dawn. I'm gonna not interfere between you and questioners. So whoever wants to ask questions, just talk to Dawn and I will listen.
Nellie, I see your hand. Oh no, that's a clap.
Dawn, I mean, I know that people started to do in vivo and to see how this motor switching plays a role. To what extent can you test some of these ideas in an intact animal? Is this possible? Sorry for these, like totally out of the. Like I was interested by this clap thing that you found. You know, like, does it change the whole network, the whole animal, and when get that activated?
Yeah, so Florian was a grad student with Wolfgang Stein and collaborated with Mikey, and he did the stimulations of the POC in the intact crab, mostly intact, and generated the movements of the teeth that replicate what we would have expected, but they weren't really monitoring anything else. Mm-hmm. Right. And at the time, that was before we knew how widely these axons projected. So I am, seeing how extensively they project. I'm really curious about what else they do. Amanda, the grad student who did that work, she likes to think about it as the hangry signal. So she thinks that when the crab is hungry, it's also angry. And so it's sort of jazzing up all parts of the animal, and maybe it's walking faster to get to food and starting to chew. But yeah, it would definitely be interesting to monitor some other behavioral aspects, but I don't know yet.
Well, if they were really sensory, that would be maybe an easy way to do this, right? If they're really in the mandibles, one could probably activate them somehow in the mandibles, and then without even like putting any electrodes anywhere, just monitor behavior or so.
Yeah, yes. I need to learn more about the mandibles. I'm fascinated by them. I thought they were just these hard things, but there's tissue in there. It's really hard to keep it intact when you have to crack through these really hard structures. But there is tissue and there are nerves projecting in there, but I don't know what types of sensory information they might be picking up. But you're right, that would be a way to externally activate them.
I would be surprised if the cell bodies were up there though, but I don't know. I'm just guessing by like insects and stuff where cell bodies are usually not out there, but I don't know.
Yeah, it's just very mysterious about where they are. I was sure they were going to be in the thoracic ganglion.
So Dawn, the early stuff we did on the club was already after we had switched from Auroritis to Borealis. Is that true? Do you remember?
So the locust certainly was. I'm trying to remember. The Goldberg paper might have been multiple species. Is that right?
There were multiple species in there because we had the club structure in Homaris and crab and maybe multiple crabs. I don't remember.
Yeah.
But it would be interesting because it has Homaris and I think even Penularis had in that paper. But I'd be curious to see about a comparison of it in Auroritis and Borealis, if you have any thought that it has anything to do with tying hunger to aggression, because Auroritis and Borealis are so different in terms of their approach to aggression.
Yeah,
like,
that's really interesting.
I mean, Auroritis is so much more aggressive and than Borealis, which is pathetic. Right? Those of you who don't know, Borealis will hide when they're threatened and Auroritis will attack. So, Patsy, do you Do you have Auroritas now?
I don't have any right now. We can usually get them. They're nasty.
They're really nasty. They're
And they
really-
can reach really far behind their backs and grab you.
Yeah, no, they're very, very aggressive. But, and we started out working with Auroritas, and I think we were still in Auroritas when we did the first paper on serotonin. But I don't remember by the time we were just, so it would be interesting to see whether both are there. And if not, to check to see what that looks like in Auroritas as well as Borealis.
Yeah, so my recollection is that the club itself is basically in all species. I mean,
It
can't
is,
just- right?
it has a different shape in each species,
Okay,
right? In other words, it's much more vestigial, I think in Penularis, and Homer, I think it's much more pronounced in the crabs.
I see.
But also that goes to, where is the whole trajectory going in lobsters? It might be, in some ways, it might be easier to do the anatomy in a small lobster.
Yeah.
Because you
But-
don't have
but
to
these-
worry about that awful thoracic ganglion.
Yeah. Gosh, that dissection is a
It's
nightmare.
just awful.
There's so much gunk. So much just yuck to get rid of.
I have a
Do
bunch of
the
small
modulators get
lobsters.
out of the CNS at all?
Are small
Like-
lobsters? How small?
Well, they've been getting bigger each year, but they're probably like this. We can get really tiny ones in the summer if we want them.
Wow,
We just go collect them.
that might be
Well,
something to
we
try.
might talk to you about that.
Yeah,
Um,
we have a permit and we know where to find them. And we have some in the lab that are like medium sized now. They're not legal sized, but they might be a good size.
That's good to know.
I have a question for you, Don. So you have some evidence, it seems, that G-cifamide activates extra IH. It modulates IH in LPG. That's what you were showing at the end. Although
Yeah,
you haven't done voltage clamp recordings yet, have you?
no, we've just measured the SAG potential and that increases.
Does G-cifamide affect any other pyloric neurons?
So it excites LP, definitely increases pyloric frequency, excites IC, VD, yeah, quite a few.
Okay. So pretty much all the ones that are excited by MCN5 are also excited by G-cifamide, or
Yeah, well.
no,
um,
actually the other way.
So MCN5 inhibits. many of
Inhibits
the
everybody
neurons.
other than
It-
the pacemakers, yeah.
Yeah, right. So it excites it's a little weird, right? So MCN5's initial effect is inhibitory. And then over time, the inhibition kind of wanes and the peptide effects take over. And so during a gastric rhythm that's elicited by MCN5, you do have really strong IC bursting, even though there is an initial inhibition of IC. So what was published, the Brian's paper that's published on MCN5, those were 10-second MCN5 stimulations. And it's dominated by inhibition, except with pacemakers. But the longer stimulations, those are bringing in excitation. I'm not sure if I got to your question or not.
No, I mean, my main question was about the modulation, because it's the first time that an excitatory peptide is shown to target anything other than the standard things that we know, right? So it actually goes after IH, which is interesting.
Yeah.
Cool.
Don, you showed like persistent sodium current, you showed other calcium currents, and do you have some voltage independent modulation also in these neurons?
So around rest, the leak does decrease, the input resistance increases, whether that is, really modulating just a leak current or it's, yeah, we haven't done any voltage clamp on it to know exactly what's going on, but that's a suggestion.
Dawn, that was really lovely.
Thank you, Eve.
And